Hi, Lida.
Hey.
Oh, that's a nice setting.
Hey.
I'm like, what's one of them since?
Cool.
Thank you.
Just make sure they just stand right here.
Okay, sounds good. Thank you so much.
And then you could probably.
Can I use this to charge just in case?
Yeah, for sure. Let me go upstairs and check on that first one and
Thank you.
peek my head in and I might be able to bring down a clicker.
Thank you.
How is it going, Candy?
We're doing good. Everything's set up, yeah.
Everything is set up?
She's just in the restroom.
Yeah, we saw here. So the plan, I want to discuss with you a little bit the lunch.
Oh, wait, let me, stop recording. Okay, now it's recording. Here we go. And then you could stay between here. That would be great.
Yeah, I'll do that.
And then there's no clicker, so you would just have to.
Yeah, that's good. So it's different.
Yeah, you can do that one, or you can click directly on it. I think that's better than actually using these. Yeah, there we go. So there we go.
Okay, it works.
Yeah.
All right. Thank you so much, everyone, for attending today's seminar. People on the Zoom and people in person. We have the pleasure, I have the pleasure to introduce Lidia Minguez Alarcon. Yes, I said it correctly, who is an assistant professor at Harvard Medical School and Harvard School of Public Health. She's a pharmacist in her education and an environmental epidemiologist who spent a lot of time thinking and doing research on endocrine disrupting chemicals and reproductive health. And recently she has expanded her research interest also on metabolic health. So she will try to join to present us like an overview of all her research projects. So we're gonna have the presentation and then we're gonna have about 20 minutes Q&As. Thank you, Lidia.
Thank you so much.
I think if you move it, it'll come back. There we go.
Okay, thank you so much, Lida, for having me, and thank you everyone for coming to the seminar. So I hope you enjoy. I'll be talking a little bit about my research on endocrine disrupting chemicals in relation to human reproductive and cardiometabolic health. So a little bit of background, infertility is defined as the inability to have a pregnancy for over 12 months of trying, and it is estimated to affect between 13 and 18 couples worldwide. Male factor accounts for 40% of these infertility cases and is defined by the WHO reference value for semen quality, and semen quality has declined dramatically over the past decades. In addition to this, diminished ovarian reserve is also prevalent among women and of reproductive age trying to conceive and have a baby, and increases as women's age increases. So the major problem with women nowadays is like the age of the first kid has been postponed. So it's very common to find women that they are diagnosed with diminished ovarian reserve when trying to conceive. So because of this, the use of assisted reproductive technologies in the fertility center has increased during the past 2 decades or 3 decades or so, specifically in the US. And given this, the fertility rates in the US, they have been inclining. So there are more babies born at the fertility center using these assisted reproductive technologies. So it is actually working. The problem is like a lot of couples, they don't have access to these very expensive fertility treatments. So identifying modifiable factors like chemical exposures, diet, drugs, and others that they can predict human fertility, fecundability, and other reproductive outcomes has become a major clinical and public health concern of importance. So chemicals of interest include endocrine disrupting chemicals. As you know, these are chemicals that they can enter our system. They can behave as fake hormones. and altering all the endocrine system. And they can be found in numerous consumer and personal care products, like children, they are exposed through toys, they are in personal care products, cosmetics. We found non-persistent pesticides in fruits and veggies that we eat like every day. And humans, they are exposed through inhalation, ingestion, and transdermal absorption mainly. So in my research, I focus on measuring non-persistent chemicals mostly, and we measure them in urine. And some examples include phthalates, bisphenols, parabens, triclosan, non-persistent pesticides, flame retardants. And the problem with endocrine disrupting chemicals is like there is, at least in the States, little regulation on their use. Even if the population, they try hard, they are still exposed to these chemicals. Even if you bring like a urine sample to a lab, they'll be able to find detectable levels of most of these chemicals in your urine samples. So that's the first problem. And the second problem is like exposure to these chemicals. They have been associated with a bunch of adverse health effects, including developmental, reproductive, immune, and also in the brain. I mostly focus my research on evaluating reproductive ones with 3 areas that we have been investigated like lately. One is whether diet might modify associations between exposure to these endocrine disrupting chemicals in relation to health outcomes. The second is whether paternal environments, they have any effect on the female partner probabilities of having a live birth. And the third one is evaluating chemical mixtures because we are exposed to a bunch of chemicals at the same time and not only one. So what we want to do when evaluating chemical mixtures is to better represent real life exposures. We mostly focus on fertility, fecundability, pregnancy outcomes, but we also measure cardiometabolic health during pregnancy because it may affect the health of the offspring as well. And the normal physiology of the pregnancy should be taken as a metabolic change, where increased insulin, higher dyslipidemia, and higher inflammation is very common. So these are some mechanisms by why these endocrine disrupting chemicals they might do their effects on our body. So it's very easy for them that they can be bind to PPAR receptors. And when these chemicals they bind to PPAR receptors, they can increase the number and the size of the adipocytes, as you can see in the slide, impair glucose and insulin signaling, and increase inflammation. And they can also bind to nuclear receptors like estrogen and androgen receptors, as well as thyroid hormone receptors, and this turns and altering the endocrine system disruption. And in general, they promote also inflammation, and inflammation is associated with a bunch of health diseases, as you probably know already. So most of the research that I did was using data from this study cohort that I will introduce a little bit now in a couple of slides, so you have a sense of what the data that we have and the exposures. So it's called the Environment and Reproductive Health Study. This is a cohort study that was established in 2004, and the PI is Professor Russ Hauser at the Harvard School of Public Health in Boston. So we aim to evaluate whether chemicals, diet, and other lifestyle factors, including stress, drugs, physical activity, they have any association with fertility endpoints in men and women, pregnancy and offspring outcomes, and cardiometabolic outcomes among pregnant women. So we enroll couples that they were not able to conceive naturally at home, and they have to go to the Massachusetts General Hospital Fertility Center to undergo either IUI or IVF treatment. So most of the couples, they underwent these treatments, but I'll say around 15% of them, of the women, they got pregnant naturally after enrolling in the study. We enrolled around 1,000 women and around 500 male partner during this period. So we collected urine samples to measure chemicals during pregnancy in women in trimester 1, 2, and 3. And we also have blood samples that we collected in these 3 trimesters in women during the prenatal window. And we also measure chemicals in urine samples during the preconception window for both men and women. We have data on ovarian reserve, specifically antral follicle counts, AMH, and other hormones for the female partners. And we also have data on semen quality parameters among men. So every time that the men, they were coming for a fertility treatment with the female partner, they were providing a semen sample, and we did like some semen analysis and have data on 5 specific semen parameters. So women, if they were able to get pregnant, we follow them until delivery, which was the endpoint of the study. And these are the outcomes that we have in the study. We also have glucose, lipids, and 2 inflammatory biomarkers that we collected during pregnancy. So I'll talk a little bit about diet chemical interactions. So traditionally, study cohorts, they were designed to evaluate the health effects of either chemicals or diet. But it was actually difficult to find study cohorts that they have data on both things. So this cohort started in 2004 to measure initially chemicals, non-persistent chemicals, but in 2007, we introduced a food frequency questionnaire and we collected diet information. Why evaluating diet chemical interactions is of importance? Because a nutrient and a chemical, they may be present in the same source. So we may be exposed to both of them in the same source. They also may target the same molecular signaling pathways, or they also share internal mechanisms of transport, metabolism, or cellular uptake. So this is the first paper that I'm going to present that we just published this year in GCM on hair mercury levels, serum omega-3, and glucose outcomes among pregnant women in the EARTH study. So this is an example of an interaction by source, because in the general population, fish is the main source of both metals, including a few metals, including methyl mercury, and also fish is rich in omega-3. So mercury, as you know, is a metal, a heavy metal, a toxicant, and it's known for its detrimental effects in the brain, among children, and also is related to adverse outcomes related to the cardiometabolic health. And omega-3 is well known for its benefits, including those to prevent cardiovascular disease. So we measure mercury in hair samples because it represents long-term exposure as hair grows 1 centimeter per month. And as a part of a pilot, we requested funds to measure fatty acids in serum samples. We publish other papers on diet chemical interactions using the food frequency questionnaire, but the problem with the questionnaires is that there are always concerns with misclassification of the exposure, recall bias, and other type of bias. So to address this concern and using previous publications, we were able to collect some money as a part of a pilot study to measure these fatty acids in serum samples. So we model hair mercury as a continuous exposure in groups as quartiles, and also as a binary exposure above and below 1 part per million per EPA reference values. And we found among these pregnant women, the high mercury levels, they were associated in most of the models with high glucose levels and impaired glucose tolerance. So we did another model further adjusting for serum omega-3 in a subset of this woman that we have data on this pilot project. And what we observed for some of the associations is that they were gone, attenuated, or no longer significant. So it's true that the sample size is lower, so we are losing a little bit of a statistical power here. But if you pay attention to the estimates, that is what we have to do when we are not working with the same sample size. Rather than seeing the p-value is paying attention to the estimates. The associations, they are gone. So we found these results super cool because yes, mercury, methylmercury, is a heavy metal, is bad for a lot of things, probably as well in relation to glucose levels during pregnancy. But actually, we can tell pregnant women that if these results are replicated in other study cohorts, they shouldn't be super worried about consuming fish because they are also exposed to the benefits of omega-3. Maybe just advising women on which type of fish is better for her, for them, those that they have low mercury, like cod or calamari, some shellfish, and try to avoid halibut that has like higher concentrations of mercury. So when you publish a paper, you also want to make good use of the results and provide with some tips to the general population. This is another paper that we published in EHP this year using data on the serum omega-3 that we obtained thanks to the pilot. And in this case, we evaluated the potential effect modification between exposure to phthalates and serum omega-3 in relation to pregnancy outcomes among women in Earth. So this is an example of an interaction by molecular signaling because phthalates and also omega-3, they can be bound to PPAR receptors. And PPAR receptors, they are involved in a bunch of things, including embryo development and placentation. So what we did in this paper was to evaluate phthalates as a mixture using quantity computation. I'll talk about mixtures in 5 minutes. What we did here was to evaluate a bunch of phthalates at the same time. So we create an exposure that was a mixture of phthalates. And we evaluated associations of these mixtures of phthalates in relation to a few outcomes. I'm only presenting here pregnancy loss, probabilities of pregnancy loss, and probabilities of live birth. And what we did with the sample, with the database, was to divide the woman in tertiles of serum omega-3. And we only found that phthalates, they were associated with higher pregnancy loss and low probabilities of live birth, meaning that phthalates, they are bad in those women that they have low levels of serum omega-3, suggesting a potential benefit or protective effect of the omega-3 on the negative associations between phthalates and these outcomes among women in the second tertile and in the third tertile of serum omega-3. Here's the thing. These women, some of them, they have been trying to conceive for years at home and they were not able to do it. So they have been taking supplements forever. So their levels of omega-3, they are actually very high. If I remember well, the levels of the median of omega-3 among these women is double compared to women participating in NHANES in the general population. This means that these no associations or potentially protective effect of omega-3 on the negative associations on phthalates on these outcomes may not be seen in the general population. So advising on intakes of omega-3 may protect against the detrimental effects of phthalates on the probabilities of having a live birth or avoiding pregnancy loss. And this is super important because, again, even if we try hard, we are all exposed to phthalates. So I'll talk a little bit now about chemical mixtures. So evaluating chemicals individually is of importance for trying to find potentially biological mechanisms that explain the associations, especially in vitro models or in animal models. And also in human populations for risk assessment, they are very useful. However, we are exposed to a lot of chemicals at the same time. So evaluating combinations of chemicals at once is also of importance. And this is a paper that we published recently in Environment International, evaluating exposure to a few phenols in relation to two inflammatory biomarkers during pregnancy among pregnant women. So we specifically evaluated BPA, BPS, benzophenone 3, that I don't know if you are familiar, but this is a chemical that is mostly present in sunscreen products. Triclosan, that it can be found in toothpastes, and 4 types of parabens. They are found in probably most of the lotions nowadays, unless specifically it is writing that is paraben-free. There are some studies showing that phenols, they can actually interact with some inflammatory processes in the body, and they can alter the production of some inflammatory cytokines. And why is important to assess inflammation during pregnancy? Because an adequate inflammation is related in a positive way to the health, not only of the mom, but also of the baby. In fact, there are some papers showing that higher inflammation is associated with adverse pregnancy outcomes, including preterm birth and preeclampsia, and also others adverse birth outcomes. So we evaluated the phenols individually, and we also evaluated the phenols as a chemical mixture. And when we evaluated the phenols individually, we observed that triclosan and a couple of parabens, they were associated with ShARP. Some of the associations, they were borderline because the sample size was not that big. What happened when we evaluated all these chemicals at once as a mixture, applying BKMR, which is a statistical method to evaluate all these chemicals at once, that we didn't find any associations with the ShARP. This method allows you to tell you which of the chemicals within the mixture outcome association is the most important one. And it shows that triclosan was a major contributor, which is consistent with individual associations. But the whole mixture didn't have any particularly effect on this outcome. And we didn't find any associations with IL-6. So as important as evaluating individual chemicals is evaluating the whole mixture. So BKMR is a very cool tool, and it can be easily applied for continuous outcomes. The problem with BKMR is that initially it was developed with continuous outcomes, and later they developed the package to be applied with binary outcomes, but it's a little bit more tricky to use, and it takes forever sometimes to run the models. So that's why some other people, to evaluate mixtures, they also apply quantile computation. And this is a paper that we published over the pandemic on exposure to phthalates, also mixtures of phthalates in relation to thyroid hormones during the preconception period. So this is a work that we did with Tim Korevar, who is a collaborator in the Netherlands. He's an endocrinologist, and it's fantastic working with him. And we applied, for this paper, we applied principal component analysis and also BKMR. And principal component analysis is actually a tool that is very basic, but it's actually very cool, because what it does is, like, you put all the chemicals in the database, and without considering the outcome, it creates components based on the correlations of the chemicals. So basically, each component represents a source of exposure. Typically, all the parabens, they are together in one component, the DHP phthalates as well. So we ran the principal component analysis for this paper that we included 10 or 12 phthalates, and we end up like obtaining two components, the DHP and the non-DHP1. And we found that mixtures of the urinary DHP metabolites, they were negatively associated with some thyroid hormones that they were measured in serum samples during the preconception period. BKMR, when we ran BKMR, what told us was that MEHHP was the primary contributor to the previous negative associations. And I'll present a couple of papers on paternal environments. So pregnancy cohorts enrollment, typically for traditional pregnancy cohorts, enrollment occurred during pregnancy. So there were mostly moms enrolled in studies evaluating exposure to chemicals during pregnancy in relation to the health of the mom and also of the offspring. But the paternal environment may be also important to evaluate during the preconception period because there are some animal studies that they are demonstrating that this paternal environment exposures that can be diet, can be chemicals, can be drug, stress, and others, they might affect the offspring health via epigenetic alterations that they are transmitted through the sperm. So when we talk about men and when we talk about evaluating chemicals exposures in relation to reproductive health in men, the first outcomes that comes to our mind is semen quality. So semen quality represents men's fertility potential. That is the primary thing that everyone thinks about. But actually, there are a few papers showing that semen quality is also a predictor of long-term health. As some men that they have very low semen quality, they have very bad sperm, they are also at higher risk later in life of cardiometabolic health and even premature mortality. So it's also a predictor of long-term health in addition to have this in addition to be a marker of fertility potential. So there are a couple of meta-analyses demonstrating declines in semen quality parameters that Shanna Swan at Mount Sinai and others, they have documented. The first meta-analysis mostly include data in Western populations, but the second meta-analysis, they also include data in other continents like Asia and Africa and South America. And they were able to prove that sperm concentration and sperm counts, they have declined over the years. We wanted to replicate what Shanna found in the meta-analysis among men in our study, including semen samples that they were spanning almost 2 decades. And we didn't not only found declines in sperm concentration and count, but we also found declines in morphologically normal sperm and in total motility among these men. So this was the first objective that we have for this paper. And we wanted to go farther, and we also evaluated because we have a huge database with a lot of variables, diet, chemical, demographics, which, if any of these variables, they were in part responsible of these declines. And what we observed in any regression for any outcome, we were adding age, we were adding BMI, education, BPA, triclosan, different types of diets. And we only found for a sperm concentration, I think, and a sperm morphology, that the decline was attenuated by almost 20% in each model when we were adding some type of phthalates in each of the models, meaning that exposure to phthalates in these men, they were responsible in part for the declines that we observed with the sperm concentration and sperm morphology, which is actually super cool. The problem with semen quality is that it's a proxy of male infertility. There are some papers among men that they have very bad semen quality, so they are considered to be infertile, but after several years, they were able to have a baby with a partner. So what we really want to know is whether paternal exposure to some of these chemicals, they have any impact in the probabilities of having a live birth or the probabilities of conceive in the female partner. And this is what we try to address here. We forgot about the semen quality, about the specific male outcome. And what we did here, what we published in this paper, or what we were trying to see was whether paternal mixtures of some of these chemicals, they were associated with pregnancy outcomes among the couples. We ran principal component analysis, and we ended up having 3 components. One is the paraben factor that typically they group together because of the high correlation. Another component was the BPA and non-DHP phthalates. And another component was the DHP factor. And we observed that higher urinary concentrations of the DHP metabolites, they were associated with higher fertility treatment failure in the female partner, even when we adjusted for female DHP concentrations, because we really want to know whether the effect comes from the mom or from the dad. So when we adjusted for the female exposures, the associations, they remain. So I guess when we publish a paper, we come up with a different question. So why we are observing that the paternal environment is associated with the female partner pregnancy outcome? It is because of changes, it is because of omics, changes in epigenetics in the sperm, in blood, because of metabolomics. And we just got this paper accepted on Monday, where we were trying to evaluate, we got a pilot study to evaluate some non-target metabolomics in male serum samples. And we were able to conduct this study with my colleague Audrey in Emory University, where we were trying to evaluate whether some metabolites or the serum metabolome in the male partner was associated with both urinary concentrations of phthalates and also couples' probabilities of live birth. And we conducted a meet in the middle analysis. I'm only showing part of the results because I don't have space in this slide to show all of them. So we conducted this meet in the middle approach to identify, as I said, metabolites or metabolic pathways that they were associated with both phthalates and also live birth. And we identified some pathways that they were involved in amino acid and carbohydrate metabolisms that may explain in part the associations between paternal phthalates and couples' probabilities of live birth. Although this is not a sperm, the non-target metabolomic was conducted in serum samples. Although there are some papers that they are demonstrating high correlation for metabolic pathways measured in serum versus semen, that is something that we also need to address. It's too much.
No, it's good.
And that is what we want to address in this first grant. So I've got a proposal a couple of years ago where we are trying to determine whether a male exposure to pesticides, non-persistent pesticides measured in urine samples, are associated with couples' probability of live birth and also a couple of offspring outcomes, live birth and gestational age. And if we find an association, we want to evaluate whether that association it is explained by changes in the epigenome, both in the sperm and in blood, or in one of them. And the other one that we are working right now, we are trying to evaluate, we already demonstrating that some of these chemicals, they have a negative effect during the reproductive years. So what we are trying to do next among women that they participated in this study, we are trying to prove or to test whether these chemicals, they are also bad or not in relation to long-term health, like cardiometabolic health. And I'll introduce this cohort study that it's called the Longitudinal Investigation of Health and Diseases After Infertility. So I call it under my name because it took me forever to have my first R1 funded. So I decided to call it like me. And this is, I try to combine, this is the study flowchart for EARTH and also for LIDI. What we are doing in LIDI is we are re-enrolling women that they participated previously in the EARTH study, and we have data on exposure and outcomes during the preconception and the pregnancy window. We are re-enrolling them that they are, most of them already perimenopausal, and collecting blood samples to measure inflammation, some lipids like cholesterol, triglycerides, and insulin and glucose. And we are also collecting outcomes anthropometry like waist circumference, height, weight. And basically the study is only a week. So we were trying to do something like easy for the study participants. And day one, we are collecting the first urine sample to measure chemicals in the midlife window, and they are also completing surveys online. Day 6, we are collecting the second urine samples to, again, measure the chemicals in this window, and we'll be averaging them. So collecting these chemicals twice is of importance because they have a very short half-life. And day 7, we are collecting blood to measure the outcomes and collecting other outcomes like the anthropometry and also blood pressure that I had forgotten. So these are the aims. We want to evaluate exposure to phthalates and flame retardants in the 3 windows in relation to long-term health. In the second aim, we want to evaluate effect modification by important lifestyle risk factors for cardiovascular disease, like diet, BMI, physical activity, smoking, among others. And the aim 3, we want to calculate trajectories in these outcomes from pregnancy to midlife and evaluate whether exposures to these chemicals during the preconception and the pregnancy window is associated with any of these changes. We'll be conducting mediation analysis by fertility markers, and also we will be applying chemical mixtures, not only BKMR, but also LKMR, that what does, it'll tell you which specific window of exposure is the most important one in relation to the outcome. So what we are trying to prove in this cohort is that these chemical exposures, they are not only bad during reproductive years, but also they can be bad long term. And I'm finishing now, and I would like to acknowledge all the team members in my study. I moved to Boston 12 years ago to do a postdoc. And I grew not only professionally but also personally, and it was in part thanks to them, and also thanks, of course, to the study participants and the funding. Thank you.
That was an amazing presentation. So many stuff to present. Yes.
Gonna breathe now.
So thank you so much, Lidia, for
Thank you.
presenting all this great work and the different studies. So I guess now the floor is open for discussion.
Yeah.
Any questions, comments? We may have some people in the chat, so let me just check. So questions for Lidia?
Good question.
Okay, go ahead, Anna.
But I thought that was very interesting when you were showing the results of the sperm quality. And so once you use the chemical as, one of the ways that you're analyzing that, do you, so my question maybe should be more like, do you think that aging, what is the impact of aging in this whole process? Because we know that aging already will impact the quality of the sperm.
Yeah.
So I know that you probably were evaluating people that will be inside of maybe a reproductive age, like
Yeah.
window.
Yeah.
But do you think that aging, like getting older, because nowadays everybody's getting married also later, you know, that's a normal thing that's happening in our society.
Yeah.
Do you think that that would affect also?
Yeah, so the major predictors for infertility is age, specifically in the woman. Like 50 years ago, women they were at home and they were having babies with 20 years old, and nowadays with 40 years old because of the incorporation of the woman to labor, and that's the major predictor. But also all these lifestyles they are also related. But aging for sure.
Yeah, but
That's
do you feel like there's any, maybe would be more severe the impact from this sperm quality because of age and exposure? Would that impact?
Yeah, the thing is like maybe for the trends in semen quality, so it's not that a participant provided a semen sample in 2000 and then in 2017. So they provided a few semen samples, but basically that study, it should be taken like a ecological study where we put together all the semen samples of all the men, but it's not that we were following the men for 17
Yes.
years to see that they were aging and the declines.
Yeah.
So it was like different men that they provide two
But
samples
just
in
in
2000.
different times that you have
Different people.
that have, in this, I kind of understood that, but it's really like, do you guys have extend that for older people? Like you have some extension for aging and the impact of the chemical to be stronger on them, for example, or there's something that's more dependent.
We evaluated a stratification by age in women because the declines in anti-mullerian hormone in women is super strong after 35. But actually men, they're actually a little bit more fertile than women. And they have actually some men with 40, 45, even 50, like good semen quality. But it'll be important to see. We did the stratification by age in among women, but not that much among men. That'll be interested to see the potential relationship between age and adding.
More severe.
Yeah.
So these are the questions and then we can answer them live and then they'll know that it's being answered right now.
Okay, so we have two questions in the chat. We have one question, have you also looked to cancer risk in these populations? And then we have another question from Brittany. Hi, thank you so much for this talk. I'm wondering about phthalate AMH results. We know that AMH is not the best predictor of a woman's fertility, especially among those with PCOS. Were you all able to look at PCOS or consider PCOS?
So thank you for the questions. I already forgot the first question.
Cancer outcome.
Yeah, we didn't look at cancer outcomes, although in the midlife window, in the women that we already recruit, we have around 20% that they have breast cancer.
20% of women have breast cancer?
And that is something that we have in mind that we would like to explore in the future.
Okay.
So the study didn't focus on cancer because it included like younger couples, but now in this midlife window, we should be able to be more participants that they have cancer. And breast cancer is something that eventually we would like to explore. And the second question, one of the limitations of this study cohort is the information that we have on PCOS because we don't have very good data on PCOS, as the variables that we have for PCOS is suspected PCOS or not suspected PCOS. To have the PCOS, you also need like a reproductive hormone test. And we didn't have it for some of these women. So that is why we didn't consider PCOS as an outcome in most of our papers. We have some data on ovulatory disorders that we added always in table 1, but we don't use the PCOS that much. But I agree that it'll be super interesting to take a look of this, especially there are papers showing that these endocrine disrupting chemicals, they are associated with PCOS probabilities in women.
Great, thank you, Lydia. Other questions?
I saw in your paper about the hair, mercury and the pregnant outcome. Have you ever measured other heavy metals in here? And I saw also you have some mixture analysis. Have you applied mixture analysis on these?
No, that's a very good point. I would love to do that, but we only have a few additional metals like cadmium, arsenic in a very, very small sample size. So traditionally, they measure the mercury in the hair samples, and I think the lab started evaluating more metals only recently. So in the last batch of hair samples that they sent, they also measure more metals, but I think the sample size is very small to do. But for sure, super interesting to evaluate for sure, especially chemical mixtures. Thank you.
Presentation. Oh, our department previously did a study called the Truth Berry study. But this was looking at prenatal exposure for lead. So I was thinking of the pathways, because children surrounded from lead smelter, their baby teeth were collected and lead was found in each tooth. So this study was conducted by Dr. Joe Johnston, but this shows how pregnant mothers, when they're exposed to lead, I think it was the second or third trimester, the body mistakes lead as calcium. So that's how the lead is incorporated definitely into the human body. So for endocrine disrupting chemicals, is there like a lifespan, or is it sort of like it's just a very long time that they persist in the body? Like is there anything?
The problem with these chemicals is like they are non-persistent. So you are excreting, if you are exposed, you'll be excreting them within 24 hours after exposure. But the bad news is like if you are using a lotion, I'm assuming that you shower every day and you put the lotion every day. So the exposure becomes like chronic. So yes, you are excreting the chemical, but you are applying the lotion every day, or using your perfume every day, or eating veggies that you are exposed to pesticides every day because that's your diet. Maybe not every day, but frequently. So that's the problem with these chemicals.
I have a weird question. So, how would you rank those EDCs in terms of their toxicity? So, my question will be, which one will be the worst? Or we will try to avoid in the first place.
We saw a lot of negative effects with the phthalates, especially the DHP ones. I don't know in the talk if you saw it, we didn't find super strong associations with the BPA and the BPS and the bisphenols, for example. The benzophenone 3s, we published actually a paper that we demonstrating that had like a positive impact on pregnancy outcomes. And we were like, how is this possible? And our hypothesis was like, you are using sunscreen when you go outside and you run, or when you are exposed to the sun. So you are taking other benefits that you are not taking into account when you are evaluating exposure to BP3 in relation to pregnancy outcomes. So in this study cohort, the worst, the phthalates. But I mean, I don't know. So we didn't find like super strong associations with the phenols, but in other groups they did. So I'll say phthalates. Thank you so much for your question.
Okay, so we have some more questions in the chat. I'm gonna just read them. Henry Barnes, thank you for your presentation. Did you explore endocrine effects beyond the outcome of infertility due to chemical exposure? For example, did you tie the DHP and paraben concentrations in men to decrease in testosterone levels? So let's start with this, and then we have some questions from Martin.
Yeah, I didn't do it, but some of my colleagues, they evaluated testosterone years ago. And I think they were able to prove that some phthalates, they were associated negatively with testosterone levels. So there are some papers out there, and I think in this study at the very beginning, before I joined, they did it. Yes, in the EHP paper that we published, the second one that I show on mixtures of phthalates, omega-3, and pregnancy loss, they also told the reviewers when we submitted the papers, they also told us if we can further adjust for methylmercury because it could be there could be some residual confounding. And so yes, we did that. I think it's as a supplemental material if you take a look at that paper.
This was a question from Martin.
No, very nice talk.
Huh? Okay.
Yeah, that was a question from Martin and this.
Okay, so have you tried to analyze simultaneously methyl mercury, omega-3 and folate? Okay, great. Any other questions? Yes.
A question regarding the study design. So I think there's a recent paper by the Alcohol Consortium talking about some of the chemicals we discussed, the flame retardants. And then there's an association shown that exposure of those type of flame retardants leads to elevated risk of autism. I'm just wondering, because you collect so many good pregnancy exposure, and then do you have plans to expand your study to collect like childhood outcome and then see like pregnancy is a kind of critical window for a lot of childhood development disorders, so I just wondering.
That's a fantastic question. Yes, one of my advisors and a colleague in Brown University, they are both co-PIs of a grant that is that aims to evaluate maternal and paternal exposure to phthalates in relation to neurodevelopmental outcomes in the children that they were born in Earth. So they didn't evaluate specifically flame retardants, but the data is there. To have someone who has interest in the topic to conduct the analysis. It's a very good question. We just have to sit down and answer that. Yeah, thank you.
Okay, great. I have a final question. I mean, for many years, we were focusing on pregnancy and childhood outcomes, but it looks like the middle life for women, it's a very important time window. The peri and menopause time period, it's a very important time window for future outcomes, for longevity, and this has been understudied so far. So your study actually targets this time window. So what will be the questions that you would like to answer with the data that you have that could impact women's health?
So when I talk about the semen quality, I thought that semen quality is also a predictor of long-term health. So I would also like to prove in this study cohort that women that they have low ovarian reserve or low levels of AMH, they also have worse cardiometabolic health during midlife. And that can be tied to the chemical exposures because maybe the chemicals are the ones that they are causing all this.
Okay. Nice.
Thank you, bye.
Thank you, Lidia. Thank you, everyone, for joining us online. We're gonna close this and stop sharing. Thank you so much for attending online, and thank you so much for attending in person. And I think we have lunch outside, right? Yes, we have. Okay. Thank you so much, everyone.